My research was about Osteogenesis Imperfecta, and I was supposed to have Isra on with me, but she's currently in SEO, and she can't be here with me right now. So we did bioinformatics at BCC in the chemistry department, and our mentor was Dr. Brennan, and our co-mentor was Professor McMahon. And to start it all off, what is bioinformatics? Well, bioinformatics is just compiling a bunch of human genomes and other genomes into the computer, to organize it into a big library and such. And what NCBI is a big part of that. I think it's National Center for Bioinformatics or something. So they're really a big help. Bioinformatics with human genetic mutation and variation, which is there is genetic diversity between all humans, even identical twins. This can be proven, some identical twins have different thumbprints, which, it's important. Human genetic mutation is when there is a change in the DNA itself, while human genetic variation is when there are differences in alleles, adenine, guanine, and all that, so it's A, C, C, G, C, A, C, whatever, like that. This is just a slide of DNA. Now, how does DNA code for protein? I hope everybody knows. It's DNA. They use DNA is inside the nucleus, so RNA polymerase. It codes the DNA into RNA, and then messenger RNA is carried out of the nucleus by ribosomes, and then ribosomes code the mRNA, transcriptive RNA, into tRNA, and then the tRNA is coded into amino acids, which are coded into proteins. And now, osteogenesis imperfecta. It's a hereditary disorder that causes bones to be very fragile and break easily. There are many other causes, there are many other things that come with osteogenesis imperfecta because the bones are so brittle that most people with it have very bad hearing because the small bones in their ear aren't formed properly. So many people have hearing aids and such. It also causes small stature, scoliosis, bowed legs, which are legs like cowboys. So it's like this. They walk like this. All right. So there are different types of this disorder. It starts from OI type 1 through OI type 8, and each type they're similar, but they have their own specific differences. And the history behind it is, osteogenesis imperfecta has been recorded over various periods in time, such as Egyptian mummies. There's one Egyptian mummy that was found to be around 1000 BC that had osteogenesis imperfecta, and there was also documents that the Norse king Ivar the Boneless also had osteogenesis imperfecta. Well, that's what his name was about. And the different types have been discovered during the late 19th century and 20th century, so it's the late 1800s to early 1900s, which is where all the different types were discovered. And the mode of inheritance, well, each type has their own different mode of inheritance. It's dependent on which type of the disorder the person has. So types 1, 2, and 6 are autosomal dominant, which means that it's a 50-50% chance of the person having osteogenesis imperfecta. It's like the Punnett square. Oh, yeah. So 5 and 6 are yet to be identified because I don't know. 5 and 6 are just very, very weird. Oh, look, this is supposed to be 4. I made a mistake. OI types 3, 7, and 8 are autosomal recessive, which means they have 25% chance of a person getting it, or they need 2 of the same allele to get that, to get the type of osteogenesis. Osteogenesis imperfecta, the genetics. It's caused by 1 or 2 mutations and the COL1A1, which is collagen 1 alpha 1, collagen 1 alpha 2, CR-TAP, which is it's a collagen assisting protein, and LEPRE1, which is the leprechaun gene, leprechaun enzyme gene. And collagen 1 alpha 1 and collagen 1 alpha 2 are collagen coding proteins, while CR-TAP is an assisting collagen protein, it just said. And the leprechaun and the CR-TAP mutations are inherited recessively. Depending on which gene is mutated, a certain type of osteogenesis imperfecta is exhibited. Mutations in collagen 1 and collagen 2 result in osteogenesis imperfecta types 1, 2, 3, and 4. Mutations in the CR-TAP gene result in osteogenesis imperfecta type 7, and mutations in the leprechaun gene result in osteogenesis imperfecta type 8. In osteogenesis imperfecta 5 and 6, the mutations are still unknown. Osteogenesis imperfecta types in 6 are the rarest types of osteogenesis imperfecta, where OI types 5 occurs in 5% of all osteogenesis imperfecta births, and OI type 6 in less than 5% of all osteogenesis imperfecta births. And these are just the locations of the certain genes where the mutations are. So this collagen 1 alpha 1 is in chromosome 17 at position 21.33, which is right there. Collagen 1 alpha 2 is located at chromosome 7 at position 22.1, right here is black. Leprechaun 1 gene is located on chromosome 1 at position 34.1, right there. And the CR-TAP gene is located on chromosome 3 at position 22.3, right there. Yeah, so osteogenesis imperfecta is not gender nor culture specific. Types 1 and 4 are exhibited the most, and 1 in every 20,000 births are a birth is osteogenesis imperfecta. The different types, it goes into more specifics, but for generalization, 1 in every 20,000 births, a person has some type of osteogenesis imperfecta. Like this one right here is type 1. You see the bones are all wormy and disfigured. And this is type 4, also the same. This is a newborn, and this is, I'd say like, I don't know, like 2 months, 3 months, probably even more.
So 1 year.
So here's a chart. The clinical features of OI, which is C1, right here. Where is it? Multiple fractures of ribs, mineral caliber, malalization, which is like the bones in the neck are very like not structured properly, and just basically it's all fragile, thin. They usually have short statures and such and such. And here's another one, the radiographic findings, which are like X-rays of osteogenesis imperfecta by type. So severity of 1 is mild, severity 2, fraternal lethal, which is means like on osteogenesis imperfecta type 2, that means that the child could die from it. Type 3 is severe, type 4 intermediate, 5 intermediate, 6, 7 intermediate. 8 was just recently, is now recently being looked more upon. So the skulls, Bormanian bones, underalization, plaques of calcification, Wormian bones. It's usually Wormian bones means the bones are all like wormy, like they're curved. They're not supposed to be curved, they're supposed to be straight. Hope you all know that. And this is an X-ray, and I don't know if this is the fibia or the tibia, and it's the X-ray of the left leg. There's a curve right here, which is an osteogenesis imperfecta. See how there's no fractures or anything, so it's not a break or anything. That's how the bone was. Oh no! Yeah, so that's how the bone was structured.